Good afternoon to all. Respected Viren sir, respected Viren sir, respected Kumar sir, respected Sonu sir, and my dear friends and juniors. Today my topic is scanning electron for microscopy, guided by Dr. Rajiv Kumar sir. Let us talk about the scanning electron microscopy. These are the contents. First is principle. Second one is the electron microscope. Third is the instrumentation. And fourth one is the working of SEM. And fifth one is the application. Sixth one is the advantages and disadvantages. And seventh one is the EM is better than the QL. And electron microscope is better than the optical microscope. These are the principles. The basic principle of the basic principle is that a beam of electron is generated by a suitable source like constant filament gun and or a field emission gun. The electron beam are accelerated through a high voltage 20 kV and pass through the system of objective apparatus and electromagnetic lenses to produce a thin beam of electrons. The thin beam of electrons scan the surface of the specimen and the emitted electrons is detected by the suitable detector. First, we are talking about the electron microscope. What is electron microscope? And the electron microscope are the scientific instrument that use a beam of highly energetic electron to examine the object on a very fine scale. This examination can be yield information about these are the four types of information which are collected, the examiner collected from the examination. First is the torography. Torography means the specimen, the object what we took to examine how it looks, what this information gives the torographic information. And the morphology information gives the shape and size of the particles, which particles we take to examine. The morphology information gives the size and shape of the particles. And the composition means the elements and compounds that object is composed and the relative amount of them. Composition information tells that how many compositions are in the element what we took to examine. And the crystallographic information, how the atom arranged in that object means if we take the particle what we not see in our eye, we can see in the microscope the particular nanoparticle. If we example the nanoparticle crystallographic information means how the atoms are arranged in the nanoparticle object. Then we can go. These are the things. We have basically in electron microscopy we use the incident beam. Incident beam means the electron beams. Electron beams what we do? The electron beam put the sample and the sample was scattering 5 types of electrons. That are the secondary electrons, RV electrons, X-ray, primary vector electrons, and the cathode density, cathodons, and the specimen current. The specimen current, the electron information which provide the current to the microscope, and the beam was incident to the sample. The samples gives the secondary electron which are the topographical information, and the RV electrons with the surface sensitivity or the compositional information. The X-ray for the thickness with the compositional information. The same as primary vector electrons with the atomic number and topographical information. These are the rays which are we are talking about build, but we are talking about the instrumentation, how it builds and what type of substances in the microscope. SEM. We are first talk about the electron optical system. First we talk about the electron bonds, condenser lens, objective lens, diffraction coil. Then we talk about the specimen stage, means the chamber where the specimen is placed, and the secondary electron detector, and the image display unit and the operating system. Disclaimer is what we need to do for better result. The electron optical system and space surrounding the specimen kept at vacuum. This is the most vital thing for the experiment. I'm sorry. There are the scanning electrons, which are the my topic, the SEM. The SEM contains electron guns, condenser lens, apparatus, scan coil, objective lens, SE detector, secondary electron detector. So the inside, so this type of SEM looks like this. And what is the block diagram as this? How it looks like inside. First, I will talk about the electron guns. We are talking about that. Electron guns produce the electron beams. Electron guns, we are typically one or two types. Thermonic electron guns and the field emission guns. Thermonic guns, we want to talk about the thermonic guns. We are around the term thermonic. Thermonic means thermal energy. Same, the things are there. The thermoelectric guns, which are most common type, apply the thermal energy to a filament. Filament was used as a tungsten metal. Filament to cause the electron away from the gun and towards the specimen under the examination. Usually made of tungsten. As we know that filament is made of the tungsten and which is the highly melting point. But the second one, the field emission gun creates the strong electric field to pull the electrons away from the atom they are associated with. The electron guns located either the very top or very bottom of the SEM and fire a beam of electron to object under the examination. It is electron gun basically is the produce the electron beam which we need. This is the electron gun. This is the voltage. We supply the current. So the bias and the filament to the filament current. With the filament here and the tungsten here. When the tungsten heated, that produce the electron beam which is going through the anode coil. Here the electron beam goes. Okay. Then the magnetic lens system. The magnetic lens system is made of condenser lens, objective lens, and scanning coil. Consider the condenser lens is the lens placed right bottom below to the electron guns and used as the adjustment weight of the electron beams as per the requirement. As you can see, the electron beams are spread out. To focusing the specimen, we have to use the condenser lens. Okay. Then objective lens. Objective lens, you bring the electron beams into the focus of the specimen, the same as the condenser lens. And the scanning coil reflected the electron beams horizontally and vertically over the specimen surface. And fine beam are the required for SEM is for better product. As we need the fine beam to place to the specimen, we need the electron beam goes through the three lens and two lens and one coil. A fine beam was required to the SEM for better performance. This is the specimen stage. This chamber, the sample chamber of a SEM where the researcher placed the specimen. Specimen means the object we took that they are examining. Because the specimen must be kept extremely still for the microscope to produce the clear image. The sample chamber must be very sturdy and insulated from the vibration. As the SEM is the very sensitivity to vibration, so we have to insulate it from the vibration and stay sturdy for the producing the clear image. The sample chamber as the SEM do more than keep a specimen still. Here the specimen chamber, and the specimen we took the specimen here. And the beam is coming from here. These are the detectors we are talking about now. The detector detects the scattering beam from the when the electron beams are going towards the sample, and then they be in the surface. In the surface, some electrons are there, and when the electron beams are fall out the specimen, the electrons are scattering from the air. The secondary electron from scattering, the X-ray electrons we have to further past we study about that. Detectors, SEM various type of detectors at the eye of the microscope. Detectors are the eye of the microscope. These devices detect the various way that the electron beams interact with the sample object. For the intense, the secondary electron, which are the overheated harmonic detectors, register is the secondary electrons, which are the electrons delocate dislodged from the outer surface of the specimen, and these detectors are capable of produce the most detailed image of an object surface. Other detectors, such as the backscatter electrons detector and X-ray detectors, can be tell researchers about the composition of the substance. These are the detectors that they are immediate detector, secondary detector, backscatter detector, which detects the beam of secondary detectors detect the secondary electron, backscatter detector detecting the backscatter electron, immediate detector detecting the X-ray and other ray of the other rays starting from the specimen. Secondary electron detector. It is used for detecting the secondary electron emitted from the specimen. The secondary electrons are attracted to the high voltage and then they generate the light and they hit the scintillator and the fluorescent substances are there. This light as they detect the photon multiplier tube PMT through the light guide. The light converted to the electron and these amplified on the electric signal, which is the working of the secondary detector. What is then the image display unit? It is mostly common, we can say, that computer. The output of the signal from the secondary electron detector are amplified through the PMT, which is the photon multiplier tube, and they transfer to the display until the forming the SEM image. The computer which is connected to the SEM microscope, SEM, is the image displaying unit. Vacuum system. It is the most vital point. If there is no vacuum, there is the vibration that occurs and the whole processor goes down. It is not working down. The vacuum system, the electron optical system of the specimen chamber must be kept at a high vacuum, 10 to the power 3 to 10 to the power 4 Pascal. High vacuum minimize the scattering of the electron beam before reaching the specimen. This is the important as the scattering of electron beams will increase the probe size and reduce the resolution. Thus, the component of are evaporated to the diffusion form. Then working. Working is the electron is simple. Working is the simply we can say that the electron can produce the electron beams by the tungsten heated the current. The electron will accelerate through the anode coil. Then the beam will travel through the electromagnetic field lenses and focus down the sample. The mechanism of deflection coil guided to the beam so that it scan the surface of the sample rectangular frame. And when the beam touches the surface of the sample, it produces secondary electron, backscatter electrons, X-ray. The emitting secondary electron is collected in the secondary electron detector and converted into the signal that can be sent to the screen, which is produce the final image. Additional detector collect the X-ray and BSC and produce the corresponding image. But how the electrons here we see that when the beam touches the surface of the sample, how they interact with the electron of the sample. Three types of interaction mainly occurs, where the electron collision, when the electron collide the electrons, then the electron pass away and scattering from the cell. That was the secondary electron, and the backscatter electron, when the electron passes through the nucleus, but there is no electron collide. That electron is the backscattered electron, and that are the abu electrons. That when the electrons collide that electron, and these electrons also they are near the cell, so the electrons also energy the internal energy also caused in this collision. So the electrons goes upward, which are the RV effect, which are the RV electrons. This is the SEM electron guns produce the electron beams. This is the anode coil. This is the magnetic lens scanning coil. The electron beam passing through there, and this is the specimen stage. So the specimen is there, and the electron beams collide with the specimen surface, and the backward electrons are detected by the backward detector, and the secondary electron are the detected by the secondary electron detector, and those are the going to the amplifier, and amplifier amplify the electric signal to and passes to the computer, which we can see the SEM image. These are the applications. SEM are the all one. SEM is the one of the most versatile technique, and can be used to. First one is the image morphology of the sample, image compositional and sound bonding diffraction, and examine wet and dry samples, view the frozen material, generated X-ray from the sample for the microanalysis, and view the map gel orientation, crystallography, and the crystallography orientation and study related information like heterogeneity. What is the advantages and disadvantages? Advantages of the SEM includes a wide array of the application, the digital 3-dimension and topographical image, the versatile information gained from the different detector. SEM is also easy to operate with the proper training and advanced in the computer technology, and associated software make operation user friendly. The students work fast and often completing SI, BSc and ADS analysis in less than 5 minutes. And what is the disadvantage? Disadvantage of this SEM start with the size and cost. The SEM was expensive, large, and must be housed in the area free any possible electric, magnetic vibration interference. Maintenance involves keeping steady voltage and current to electromagnetic coil and circulation of a cool water. It is the disadvantages of the SEM. Last question, today's question is electron microscope is better than how the electron microscope is better than the light optical microscope. We generally use the optical microscope, the light microscope, but electron microscope how far better than the optical microscope. These are the things. EM means electron microscope. Electron microscope proved to be much more powerful and allowed the scientists. I am these are spelling mistakes. To view the nanostructure, some electron microscope technique employ a resolution of 0.2 nanometer, which is more than 1,000 times better than the microscope, light microscope. With the high resolution of electron microscope are able to magnify the specimen up to 1 million times, allow the researcher to study briefly about the nanoparticle structure. How? This is the answer is region 1. In the EM, we can say electron microscope. Electron microscope is the electron beam. Starting at the electron beam, the beam of electron used, which interacts with the sample and transmitted through the specimen. As we know, this is pretty mistake. As we know, the electrons are stable and subatomic particles, so electron beams have much more shorter wavelength than the visible light. Visible light microscope, which we can consider deep-oblique wavelength, which is around 0.091 nanometer. The shorter wavelength allows the higher level of detail and resolution image of electron microscope. And return to us, we discussed about the focal microscope, which is the optical microscope. We also know that the focal microscope crop beam focused by the lens. At the problem of the lens, we have to remember. So resolution is d is equal to lambda by 2 beta. But the lambda is equal to wavelength of the particle, beta is equal to sin theta, which is the numerical apparatus. For the visible light and high numerical aperture, the beta is equal to 1 and d is equal to 200 to 400 nanometer. So the direct optical imaging not useful the nanostructure matrix study. But the optical microscope is giving good picture of nanoparticles, but for studying a detail, we prefer to use electron microscope for research purposes for the nanoparticle study. This is for today of my topic. Thank you very much.